Hi, I'm going to talk about geographic ecology and then focus on species area curves. I'm basing this on the ecology textbook by Manuel, Molles, and Andrea called Ecology Concepts and Applications. It was published in 2019 as the 8th edition by McGraw-Hill. I'm Sue Glenn, and I teach ecology at Rowan College at South Jersey. This is a topic that's near and dear to my heart. I have a Bachelor of Science degree from the University of Toronto in biogeography, and so geographic ecology is something that has been at the base of my thinking about ecology for my entire career. And I'm going to talk a little bit about the history of this field. Alexander von Humboldt is credited for actually being the modern beginning of the geographic ecology field or biogeography field. In 1977, he started on his first journey from Europe down to the Canary Islands and then down to South America. And he was traveling with a botanist, Ami Bonpland, and they were supposed to be systematically surveying the islands that they would visit or the lands that they would visit. And but he wanted to also study how the geographic environment affected plant and animal life. And so they were really doing a complete scientific survey, and they ended up traveling over from 9 to 10,000 kilometers through South and North America. They traveled by ship to get there, on foot, by canoe, on horseback. They went from 12 degrees latitude south of the equator to 50 degrees latitude north of the equator. And they climbed up to 5,900 meters, which is the highest anyone had climbed at that point. It was the mountain Chimborazo, which was the highest anyone had ever climbed, and it was really hard on them. They didn't have all the equipment we have now. They didn't know how they were going to survive the altitude sickness. And even as they were doing that, they were collecting plants and looking at their altitude and what sides of the mountains they were growing on. And it was really inspirational. Um, and following his journals, his journeys, he wrote all sorts of books and publications. He wrote more than 36 books, and had traveled to 4 continents. But he also met with a lot of people, and he was very outgoing, very charismatic. And they never knew what was going to come out of his mouth. So he appealed to a large number of people, and he influenced politicians, and he influenced artists, and he influenced other scientists, including Charles Darwin, who, when he read about Humboldt's expeditions, he basically said, "That's what I want to do," and changed his life as well, and went in that field. So his interesting personality resulted in him being remembered. Places were named after him. Animals, plants were named after him. University, there's towns. There's all sorts of tributes to Humboldt. Even places he never went to, that people were influenced by him. He said the claims are some fables about Humboldt. He claimed to sleep only 4 hours a night, and he called coffee concentrated sunbeams. Uh, this image is some illustrations by Lillian Melcher from the book called The Adventures of Alexander von Humboldt, written by Andrea Wulf. Andrea Wulf had written a really good book that was on the New York Times nonfiction bestseller list about his journals. And then, in order to increase readership, she worked with Lillian Melcher to make an illustrated version of the journals. And Melcher had taken various drawings that Humboldt had done from his journals and his writings and tried to make them come alive in these images, which I think are absolutely amazing. Don't really have the time to go into all of the details of what he had written about in all of those books, but some of the things that really were first, was talking about how the continental land masses were probably spreading through plate tectonics. He mapped the distribution of plants on 3 continents. He looked at how air and water currents moved and changed, which resulted in different types of climate at different latitudes and at different altitudes, which was going to impact the biomes, impact the plants and animals. He looked at how deforestation was affecting local climate, so he was dealing with microclimate effects as well. And he found in the geological strata fossil remains of plants and animals that he realized were the precursors to the modern forms. So really looking at extinction before other people were looking at extinction. So he had died before Charles Darwin published The Origin of Species, but I'm pretty sure he would have been really interested in reading that book. And this is another collage of some of the botanical specimens that Humboldt had observed, plus some illustrations from some of his stories of things that he'd seen happening. Some studies in the early 1900s had found that there seemed to be more species in areas that were larger than there were in smaller areas. And it was actually Frank Preston wrote a couple of papers in 1962 in the Journal Ecology, sort of looking at the theoretical underpinning of this. But it was all based, these ideas were all based on empirical data that they had. Preston had data on birds in the Caribbean, looking at the relationship, saying that the small islands had fewer species, richness, fewer numbers of species than the smaller islands had fewer than the large islands. And then a group of researchers were looking at islands in Sweden, and not just birds, but they were looking at beetles, woody plants, land snails, and really they found that the size of the island was really the largest variable that they could find. This was work done in the 80s that would actually predict how many species there would be on the islands. The next slide has some of the data from these two different groups of researchers. The left, this is showing you the data from the work by Preston, where we have the island area across the bottom. Let me just see if I can get a pointer here. So we have the island area across the bottom, and this is, you can see it's a log scale. We're going from 100 to 1,000 to 10,000 to 100,000. And then species richness, the number of species is on the y-axis. And it's very clear, you can see that the number of bird species on the larger islands has a much higher number than the number of bird species on the small islands. The bird species is also on a log scale. And then we have the work that was done by Sven Nielsen, Jan Bergsten, and Stefanos in 1988, where they were looking at islands in a large lake in Sweden. And they also found the same relationship, that the bigger islands had more species on them. So once again, these are logarithmic scales on these graphs. And more studies were finding this relationship when you looked at the data. Even though we think of islands as being a land area surrounded by water, it's really how we define an island, but we can see other habitats can act functionally as an island, where you have an area of habitat like these mountaintop areas where the surrounding hot desert is going to be too hot and dry for organisms to move across from mountaintop to mountaintop. And the way this happened was these species got here during the last glacial period in the Pleistocene, which was around 11 to 15,000 years ago. And the glaciers were across North America, the northern part of North America and northern US, and had pushed the cold-loving plants and animals further south. So this is looking at in American deserts in the southwest, there was all these forests and the animals associated with those forests. Well, when the glaciers warmed up and they melted, and this habitat started to get hotter and hotter, then those cold-loving plants and animals not only moved north, but they moved up the sides of the mountains, so they increased in altitude so that they could get up into a colder habitat. And we still find these isolated pockets of these communities up on these mountaintops that are separated by these deserts where they can't really easily jump from place to place. So Mark Lomolino, Jim Brown, and Russ Davis did a study in the 80s where they looked at data from 27 mountain islands, these mountaintops in the American southwestern deserts, and they looked at 26 species of forest mammals. So they didn't look at bats because those could fly from mountaintop to mountaintop, but they looked at the ones that would be confined to trying to walk from place to place. And they tried to pick areas that were well studied, so they knew they had good data sets from that. So if we look at this graph here on the x-axis, we have the area of the mountaintop that they were looking at shown in just highlight that, shown in square kilometers, and it's a log scale. So just like we had before in species area curves, we're looking at a log scale. In that case, this number of mammal species that they found is a numeric scale. So this one is not a log scale, but we can see a very clear correlation where the bigger areas had more species found in them than the smaller areas. So these were acting just like we were seeing with the islands in the water. This is another example of something that acts like an island, and that are these lakes in northern Wisconsin. This is in Vilas County, Wisconsin, and this study was done in the 1980s by Tone and Magnuson. And we have all these, remember I talked about these glaciers being all across the northern U.S. Well, what they were doing, this ice was sitting there and scouring holes in the rock, and all these holes in the rocks became all these little isolated or small lakes. In this particular county in Wisconsin, there were over 1,300 lakes. And so what they did was they looked at 18 different lakes that had similar characteristics. So they had similar depth and similar substrates on the bottom, but they were a range of sizes. So they ranged from 2.4 to 90 hectares. A hectare is 100 meters by 100 meters. And 10 of the lakes were drainage-fed lakes or spring-fed, drainage lakes or spring-fed, and then 8 were seepage lakes. And those 8 lakes had a history that they had low oxygen content during the winter time. Just like before, we can look at the lake area, which is shown in hectares on the x-axis, and this once again is a log scale. And then we have the number of fish species in those lakes, and this is also a log scale in this case. And just like we saw with other kinds of islands, that the larger the lake, the more species that we would find living in the lake. This relationship between the species richness and the area has been generalized. We can see this equation here. So if we have graphed the number of species as species richness, and this is an arithmetic axis, a regular axis, against the area of whatever it is we're looking at. And we can see that the number of species will increase, and it will eventually level off at some point, so that you sort of get to a point where it's a big enough area that you have sort of reached the maximum number of species that you can keep adding area, but there's no more species left in that region, so it's going to taper off. When you take the natural logarithm, or the log 10 of each of those things, so this is the log 10 of area and the log 10 of the number of species, what you do is you're pulling in some of those right tail of the graph, and you're ending up with much more of a straight line. So when you have that straight line, you're going to have an equation, y equals mx plus b. And we'll actually do a lab where we're going to figure out how many species are on different sizes of islands. And we can use this equation, y equals mx plus b, to actually estimate the parameters in the species area curve. C is a constant, and z or z is going to be the slope of this line over here. So the slope of this line, the m, is equal to the z in your equation. And it's going to be a fairly small number. It's going to be a fairly gentle slope. And then this constant at the beginning then is going to be really dependent on how quickly you start this line. So it's going to be dependent on the intercept, which is your b of this line. And we will get to that when we do our island biogeography lab with the species area curve. Area is a really important consideration in determining how many species you're going to find in a place, but you also find a negative relationship between how isolated an island is and the number of species it supports. And this is going to depend on the types of organisms you're looking at, because some are able to disperse much easier than others. So here we're looking at some islands near New Guinea in the Pacific Ocean, and we can see that the distance the islands are from New Guinea is going to make a difference as to how many species they have on them. So overall, we've got on the x-axis, once again, we've got our area in square kilometers, and we can see once again we've got a log scale. Each step on this is 10 times the amount of the previous step, and the same thing going a log scale on the number of bird species we found in these islands. So the species area curve is showing it's going up, maybe tapering off. But then if we look at a particular sized island, so let's say I'm looking at an island in this area, the number of species in the islands that are far from New Guinea, which are your red dots, are going to be lower than the number of your species that are close to New Guinea, and you get the most species closest to New Guinea. So if they are near New Guinea, they're going to be giving you more and more species. So if we're looking at distance going out in the Pacific Ocean from New Guinea, we can see it makes a huge difference. And that if you're a small island and you're far away from New Guinea, you're going to have fewer species than if you're a small island that's a little bit closer to New Guinea. So if we're going to look at the number of species as a function of how far you are from New Guinea, here we are looking at, instead of the island area, we're looking at the distance from New Guinea, and this is the percentage of the species, so the percent saturation. Remember, it sort of levels off if we're looking at the area and the number of species, it would level off at a certain point. So this is how far you are from New Guinea. You're going to, if you're close to New Guinea, you're going to be very close to the number of species you could possibly have. And the further away you get from New Guinea, the fewer, lower percentage of the total possible species has gone way down. This is again looking at the southwestern deserts that Lomolino, Brown, and Davis were looking at the mammals on the mountaintops. But in this case, they're looking at how far the mountaintops are from the Rocky Mountains, so how isolated the mountains are. And we can see that the further you get from the Rocky Mountains for those mountaintops, the fewer numbers of mammal species you find. So we can see a negative relationship as you get further and further away from the source of potential colonists coming to the mountaintop. You have much less of a chance of finding very many species. So this is really implying that, just like when we were talking about a source of individuals coming to islands in New Guinea, this is implying that there are individuals moving across between these mountaintops, and just not very many of them get this far. It's a lot harder to get to to the more distant mountaintops, but it does imply that migration of individuals from one location to the next is an important process. When I was talking about the birds coming onto the islands from New Guinea, that was work actually done by MacArthur and Wilson. So we talked about MacArthur and MacArthur's warblers way back early in the course. We mentioned MacArthur several times. MacArthur is considered a leader in the growth of geographic ecology, and Wilson had been working on looking at the influence of island area on ant populations, and they got together and started to look at this data together. And so they found that isolation reduced the bird diversity on Pacific islands. The ones that were further away from the mainland had much fewer birds on them than the ones that were close. Mark Williamson was looking at the relationship between different types of organisms, between two groups of islands. He was looking at the Azore Islands, which are way out in the middle of the Atlantic Ocean, versus the Channel Islands, which are very close to Europe. And he found that the species richness of the birds on the Azore Islands showed really a clear effect of isolation and distance from one another. It was hard for birds to get to some of the more distant isolated islands, and so you would find that the isolated ones had fewer birds on them. But he found that something different happened when he was looking at ferns. Ferns reproduce by little spores. They're very light. They catch the wind. They blow around. And the spores didn't really show any impacts of isolation when you compared them to the bird populations. So here's the data from that. We've got island area, once again, across the x-axis for a log scale there, and the number of bird species richness on the y-axis, that's also a log scale. And we've got two groups of islands. We've got the Channel Islands that are shown with yellow dots, and then we have the Azore Islands out in the mid-Atlantic shown with the red dots. And the top left one is the birds, and the bottom right one are the ferns. And so we can see when we look at the effect of isolation with the birds, those more isolated islands that were hard to get to and far they might have had the same area as a channel island, but they had many fewer species than the same size of Channel Island. So being isolated seemed to really reduce their number of species. But we can see with the ferns, where we have this overlap on sizes of islands, there's really no difference in how many species we saw. You still have the species area effect, but it's really not evident that isolation plays a role in the case. So it does depend on what species you are looking at. This is a picture in the background here of one of the Azore Islands. So Robert MacArthur defined geographic ecology as a search for patterns of plant and animal life that can be put on a map. And his map might be a group of islands or a region or a series of continents. And so obviously he was very interested in geographic ecology and was, someone who was also inspired by explorers like Humboldt and Darwin and Wallace. And he certainly, when he got together with E.O. Wilson to talk about their models with island biogeography, made a huge lasting impact. So E.O. Wilson's studies on ants led to this theory of island biogeography regarding distance and island size and isolation. And he had found with his ants that every 10 times increase in the size of the island had led to a doubling in the number of ant species, but it also depended on the distance from the source. So Wilson teamed up with MacArthur in 1963, and they made the theory of island biogeography. So the next video is going to go over the theory of island biogeography in some detail and explain how it works. It was published originally proposed in a paper published in Ecology, and then in this little book by Princeton University Press called The Theory of Island Biogeography. So at this point, if you go back over this first part of chapter 22, you should be able to see how information about habitat fragmentation and what we're doing for clearing forests and causing little isolated patches of habitat might be affecting isolation and might be limiting the ability of species to replenish populations in these little patches. So it is something to think about when we're moving forward and talking about the impacts of this theory of island biogeography, not just on understanding geographic distribution of species, but also in understanding conservation. So hopefully see you at the next lecture on the equilibrium model of island biogeography.